Welcome everybody. I'm very happy to give a presentation on the OHBM 2022, and I want to speak about our approach to move forward from correlational approaches to meta-analytical symptom reading in single subjects. First, I want to give you an overview of my talk. I want to start with the individual patient in clinical settings. Second, I want to discuss how we can increase evidence with meta-analyses. Thereafter, I want to show you how we can develop disease concepts with meta-analyses. And finally, I want to discuss how we can translate meta-analyses into clinical routine. Let's start with the individual patient in clinical settings. I just want to present you a case. This is a 51-year-old male real estate lawyer. He shows a little bit changes in his personality and in his behavior. So he spends most of the day at work shuffling papers, reading magazines, downloading pornography instead of working, which is obviously not an adequate behavior in working surroundings. Moreover, he embezzles money at work, and however, when I asked about his future, he's very optimistic, making lots of money. He loses his interest in his wife and children, watches TV instead of speaking, develops a strong desire for potato chips, and his manners deteriorate. He eats with hands, stuffs his mouth. We have deep changes in personality, in behavior in this person. If we go to international diagnostic criteria for neurodegenerative diseases, we can obviously see that this person is suffering from so-called behavioral variant frontotemporal dementia, or BV-FTD, which is characterized by several criteria. First criteria are disinhibition, and moreover, there are apathy, loss of sympathy, empathy, hyperorality and dietary changes, and finally, neuropsychological executive deficits, which is extended in the DSM-5 by social cognition. In this patient, we could observe all of these related symptoms. These are the criteria, the clinical criteria for possible BV-FTD. One can also involve imaging criteria which increase the validity of the disease and is called then probable BV-FTD. So here are the changes in frontal and anterior temporal regions of highest importance. In the next part, I want to discuss with you how we increase evidence with meta-analyses. The last decades have seen an exponential increase in publications on brain functional MRI, as shown in light blue color, and also in dementia and MRI, as shown in dark blue color. So we have a lot of information. And this information can be made available and utilized with big data mining. So we can use meta-analytical techniques as used in these different algorithms to understand diseases and understand treatment options and to understand and reach a better treatment for individual subjects. Starting from these case reports, which are the classical approaches, by the way, to neuropsychology, we can increase evidence by going to group studies or going to multicentric studies, and last but not least, going to meta-analysis, which summarize all the evidence obtained in group studies. So across this road, we can produce a very high statistical power, and this statistical power generates generalizability of our results. How can we use this information? Firstly, we can develop concepts with meta-analyses, which I want to show you in the next part of the presentation. On the left, you can see our results for meta-analysis eliciting the anatomical correlates of behavioral variant frontotemporal dementia, already discussed before. I think this is the first study, by the way, to use anatomical likelihood estimates in this context. And obviously, which is very interesting, you see that the frontomedian cortex is involved and additionally the anterior insula. What makes these results so interesting is that this network seems to be disease specific. As you can see on the right side, this network is specific for behavioral variant frontotemporal dementia, BV, FTD, in comparison to other neurodegenerative diseases such as Alzheimer's disease or primary progressive aphasia, which mainly affects language functions. If we discuss the results in the context of cognitive neuroscience, it is obvious that the frontal cortex is involved. This brain region is a rather new achievement in evolution, and humans are characterized by a relatively very high volume of frontal lobe in comparison to the whole brain. Histopathological studies have shown that these brain regions, so the frontomedian area and the anterior insula, also hotspots for von Economo neurons. These neurons are discussed to be involved in socio-emotional functions and have been shown in other highly developed species like apes or whales. If we go to histopathological studies, it is evident that in frontotemporal dementia you have a reduction in the density of von Economo neurons in these areas, which makes this disease also an interesting model disease for changes in socio-emotional abilities. Now I have shown you how we can use imaging data for structure and function in diseases. However, we could also combine this information from diseases with information from meta-analysis in healthy human subjects, which investigate cognition in healthy subjects. Here I want to present you the results of a combined meta-analysis, which uses the potential to combine meta-analysis on subjects suffering from a disease and healthy subjects. In a first step, we identified the neurocorrelates of frontotemporal lobar degeneration. Here we involved a number of 800 subjects, and we could identify separately the neurocorrelates for FGG PET, for glucose utilization, as shown in blue, and structural changes as shown in red, and analyzed with structural MRI. In a second step, we calculated so-called behavioral domain profiles. In this approach, we go to the probabilistic functional brain atlas of the BrainMap database, which contains functional MRI study results of 40,000 healthy subjects and 3,000 experiments. And here the cognitive functions are classified in several categories and even single paradigms. So we can use the neurocorrelates of the disease as already identified as a region of interest and extract the cognitive functions which are related to these neural correlates. So in the result, we receive the behavioral domain profiles, and these extract behavioral, psychological, or mental correlates of networks involved in behavioral variant frontotemporal dementia. The analysis was done across the 5 cognitive dimensions and 83 paradigm classes as contained in the BrainMed database. In the result, we could identify with this data-driven approach networks which are related to emotion processing with 5 clusters, to reward processing, these are again 5 clusters, and additionally cognition, inhibition, and pain processing. In the next slide, I want to show you the correlates separately in blue for hypometabolic regions and in red for the atrophic regions as identified by FDG PET or MRI. And here you can see the regions which show a significant association with emotion, mainly what I have discussed before. Also cognition, here cognition, for instance, the so-called inferior frontal sulcus inferior frontal junction area on the left side. Finally, we wanted to validate the behavioral correlates of the disease. So we performed conjunction or overlap analysis. We took on the one side the neurocorrelates as is extracted in the first step for behavioral variant frontotemporal dementia, and we calculate additionally meta-analysis with activation likelihood estimate method in healthy subjects. Why have we additionally used meta-analysis for other cognitive domains? Because these domains were not contained in the BrainMap database. Domains of interest were especially empathy and theory of mind as a proxy for social cognition. Here you can see the conjunction analysis, again in blue, the changes in glucose metabolism, and in red, the atrophy, and empathy in green, and theory of mind in blue. And you can see that in the frontomedian areas, as shown in yellow or here in white, we observe an overlap between these cognitive functions and the disease. We have done the same for moral cognition, here shown in green, and again, the frontomedian cortex seems to be the central structure involved. Why is it interesting to investigate moral cognition? Because subjects with frontotemporal variant dementia show 40% of cases also criminal behavior, which makes it a very interesting disease and is also a clinical symptom of the disease. In the end, as shown on the right side, we developed a concept which you can read in our papers, and the central structure was here the frontomedian cortex, which unites the neurocorrelates of behavioral variant frontotemporal dementia and the cognitive dysfunction. We also validated our concept with correlational approaches in our own patient cohort for dementia syndromes, and as you can see here in a correlation analysis between changes in glucose metabolism and brain function, there was an overlap for apathy, disinhibition, and changes in appetite and eating behaviors in the frontomedian cortex. So obviously the frontomedian cortex is the central structure involved in BV, FTD, and relevant also for its clinical symptoms. In the last part of my presentation, I want to discuss how we can translate these meta-analysis into clinical routine. I started my presentation with a case report of a patient suffering from frontotemporal dementia. By the way, this is not the patient treated in our department, but it's Friedrich Nietzsche, who was discussed to have suffering from frontotemporal dementia. So we want to mirror our results back to the society and support individual subjects. Before I have discussed that doing meta-analysis, we increase statistical power and increase also generalizability. But on the other hand, we lose personalization or subjectivity. Therefore, we have to return from meta-analysis, from a quantitative approach to qualitatively subjective patients to improve diagnosis and treatment of these subjects. Here I want to show you some results, what we obtained with support vector machine classification and structural MRI data. We classified behavioral variant frontotemporal dementia in comparison to controls. Here you see the results. And we reached an accuracy in classification with support vector machine classification across the whole brain of 82%, and we could increase the accuracy if we focused on frontotemporal regions of interest as elicited in the meta-analysis. So we could improve our diagnostic accuracy by using meta-analytical results. In the result, we can support diagnosis of single patients with meta-analysis and machine classification. Finally, I want to show you that we cannot only predict the syndrome or the disease in these subjects, but we could even predict symptoms in single patients. So I want to go to another case report. Now I leave neurodegenerative disease and go to stroke subjects. This is a 56-year-old woman with bilateral stroke lesions in the so-called inferior frontal junction area. You can see here in the MRI image the lesions, bilateral lesions in this area in the frontal cortex. This was the so-called middle cerebral artery territory. In the right figure, you can see the neuropsychological profile of this patient in comparison with the healthy control cohort. And you can easily observe that we have a specific change in executive function. So this patient is heavily impaired in executive function. This is very interesting because the inferior frontal junction area has been related to executive functions according to meta-analyses and also case studies. We have now developed another tool, also based on behavioral domain profile analysis. This is so-called meta-analytical reading of symptoms, or MARS. So we might use this as MARS attacks, our new method. Here you can see again the lesions illustrated in the upper slide for the patient in the inferior frontal junction area. This lesions affected network as revealed by meta-analytic connectivity modeling across the brain, and in the lower part you can see the results of the behavioral domain profiles. And interestingly, behavioral domain profiles revealed that the lesioned areas were mainly related to executive functions like Stroop color word task, memory, working memory functions, also NBAC task switching, but additionally to language functions. It is quite interesting that this patient also showed, when we investigated him, executive dysfunction, but in the beginning language function as well, which disappeared in the meantime. So let me discuss the perspectives of our results. Meta-analytical data mining reveals neural networks facilitating diagnosis and differential diagnosis. We can reconceptualize neuropsychiatric diseases with these meta-analytical techniques, and we can personalize medicine if combined with machine learning. So let me just give you a perspective what lead from on the road from diagnosis of neurodegeneration to therapy inducing neurodegeneration if we use meta-analytical tools. I call it the magic black prediction box, because we have several methods which we can use here. And I think that meta-analytical techniques can, together with machine learning techniques, contribute a lot to personalized diagnosis, differential diagnosis, symptom prediction, also histopathological prediction if combined with PET techniques, and personalized therapy approaches. And even we could predict prognosis if we translate meta-analytical techniques together with machine learning to everyday medicine. Thanks a lot for your attention. And now it's time for questions or comments if you have any. Thanks a lot.